So I'm Sofiana Torres Palacios, and I'm going to talk about animal and plant cells, organelles, and its functions. First, I'm going to talk about the animal cell and its organelles. We have first the cell membrane, that is a selectively permeable membrane that controls what enters and exits the cell, acts as a fence around the cell. The nucleus, that is the largest organelle in an animal cell, contains the cell's DNA and chromosomes, coordinates major cell processes like DNA, replication, transcription, and translation. The cytoplasm, a water-based solution that allows organelles and substances to move around the cell. Proteins, organelles, and other cell structure float in the cytosol. For the endoplasmic reticulum, that is a network of membrane that processes and transports new materials. There are two types of endoplasmic reticulum, the rough endoplasmic reticulum and smooth endoplasmic reticulum. Fifth, the mitochondria, known as the powerhouses of the cell, break down glucose into adenosine triphosphate, known as ATP, which fuels other cellular processes. Sixth, the Golgi apparatus modifies proteins and lipids and transports cellular substances out of the cell. The ribosomes, organelles that synthesize all proteins in the cell. There can be as many as 10 million ribosomes in a single animal cell. The lysosomes are round organelles that contain digestive enzymes that help with digestion, excretion, and cell renewal. Ninth and last of the animal cell is the vacuoles that stores and dispose of various substances, including food and other nutrients. Now, the plant cell. The plant cells have many parts that perform specific functions, and we can know many of them because of the animal cell, because they're kind of similar. First, the cell wall that provides structure and protects the cell from mechanical stress. The chloroplasts contain chlorophyll, that is a green pigment that enables photosynthesis, which produces glucose and oxygen. The central vacuole that stores water, helps maintain pressure, and aids in waste disposal. The mitochondria, that I already said, break down glucose and sugar molecules to produce energy. The nucleus houses genetic information and controls cellular activities. The endoplasmic reticulum transports proteins and lipids within the cell and also acts as a plant sensor. The Golgi apparatus processes and sorts proteins received from the ER for transport to their destinations. The plastis, that is a double membrane organelles that manufacture and store food. The ribosomes, organelles that help in protein synthesis, which is required for many cell activities. The chromatin, that is fine-tunes the accessibility of DNA and modulates gene expression. The cytoskeleton helps cells maintain their shape and internal organization and also provides mechanical support. And last, the plasmodesmata facilitates communication and material exchange between cells.
Hi, today I'm gonna talk about characteristics of living things. How do we know that something is life? I mean, this answer seems obvious and intuitive, but when you ask that question in the context of searching for life in other planets, it becomes more difficult to pin down. In the unfamiliar environments of our planets, we need to find a way to distinguish between a phenomenon that isn't alive compared as one that is. One way to address this is by considering the characteristics of all living things, which reveals how much all living things have in common. Because life is so diverse, it stands to reason to think that it's impossible to define it. However, seeking a definition of life, most biologists have concluded that there are 8 key characteristics that all living organisms share. Number 1. They are made of one or more cells. The cell is the smallest unit of life, and all living organisms are made of cells. Number 2. They have metabolisms. This means living organisms are constantly degrading substances and replacing them with newly manufactured ones. Number 3, they maintain homeostasis. For the metabolic process to occur correctly, living organisms must keep several critical factors stable, such as temperature and acidity. Number 4, they respond to stimuli. Any living organism can perceive and react to changing conditions themselves. Number 5, they possess DNA as their genetic material. DNA, as all we know, contains all the information required to control the various processes and keep an organism alive. Number 6, they grow. They increase in the number, size, and volume of cells. Number 7, they reproduce themselves. This ability is determined by the genetic material contained in the cells. And finally, number 8, they evolve. This means all living things changes to better adapt to their environment. Now let's talk about cell types, the prokaryotic one and the eukaryotic one. First, cells can either be prokaryotic or eukaryotic. Each type is different in size and the type of organelles they contain. The prokaryotic cells, meeting before the nucleus, are found only in the bacteria and archaea, while eukaryotic cells, meaning through nucleus, form the structure of protists, fungi, animals, and plants. Now let's see some differences that they have between each other. First, the prokaryotic cell, that is smaller with 1 to 10 micrometers. It doesn't have a true nucleus, the lack of membrane, has circular DNA, and it has a sexual reproduction. And the eukaryotic cell is larger with 10 to 100 micrometers. It has a true membrane, contains membrane-bound organelles, has the DNA, linear DNA contained within the nucleus, and it can reproduce sexually.
Hello, my name is Fernanda Redondo and I'm going to talk about the cell theory and the history of the microscope. The microscope was first invented in the late 16th century by Dutch sparkle makers Hans Janssen and his son Zacharias. They created the first compound microscope using two lenses of magnify objects. In the early 17th century, Anthony Van improved the microscope and was the first to observe microorganisms like bacteria and protozoa using the single lens microscope. His discovery marked a beginning of the microbiology. In 1665, Robert Hooke used a microscope to observe cork and discovered small box-like structures which he named cells. This was the first use of the term cell in microbiology. In the 19th century, the cell theory was developed. Matthias said then in 1838 concluded that all the tissues are made of cells, and Theodor Schwann in 1839 extended this idea to animals. Together, they established the first two parts of the cell theory. The first part, all living things are made of cells, and second part, the cell is a basic unit of structure and function of organisms. In 1955, Rudolf Virchow proposed the third part of the cell theory, that is, all the cells come from pre-existing cells. These discoveries, made possible by the microscope, transformed our understanding of biology, revealing that cells are the building blocks of all living organisms. The microscope remains a crucial tool in studying life at the microscope level.
Hi, my name is Hector, and I will be explaining homeostasis and levels of organization. Homeostasis. One of the main characteristics that distinguish living things from non-living things is the ability to maintain their internal conditions despite the conditions of the external environment. This is known as homeostasis. is a physiological system of processes constantly measuring and compensating for changes in the outside world that could damage our internal systems. What does this mean? Homeostasis is a process that happens inside of our body that protects us from changes in the environment or external world that can damage us. For example, humans have the ability to sweat that regulates our internal temperature, making us the best species in the world. Now we'll go to levels of organization. First of all, when we talk of organization, we rapidly think about something looking organized or an event super organized. But talking in terms of biology, we encounter that, for example, for simple and normal things we, the humans, do, like maintaining glucose levels, maintaining a good and healthy temperature level, a proper level of oxygen in blood, and either performing different and multiple tasks simultaneously. We need organization. For all, we need first organization. One example of organization in nature are molecules. You may be asking, why molecules, if it's only a combination of some elements and that's it? Well, molecules are not as simple as that. For example, for creating a molecule, we need the proper size of the molecule. Hundreds of thousands of millions of carbon, hydrogen, oxygen molecules. Then, that molecules can convert into organelles. That is the basic thing of a cell, the fundamental unit of living things. For example, us, dogs, cats, horses, etc. Then, after creating that cell with millions of that, it can be created tissues. The tissues, in simple words, are a part of an organ. And organs, as we know, make systems that make us function correctly. In animals, there exist 4 different types of tissues: the connective one, the epithelial, the muscle, and the nerve one. The connective one connects other tissues like fat, bone, blood, and lymphatic tissues. The epithelial one, it maintains function. Its main function is to cover our internal parts, such as the skin. Muscles consist of striated muscles that help us move, and finally, we have the nerve tissues that helps us feel things.
Hi, my name is Carlos Cifuentes, and I'm going to talk about pregnancy, obesity, and addictions. Pregnancy is a crucial time, but addictions and obesity can put both mother and baby at risk. Smoking, drinking alcohol, and drugs can cause premature birth, low weight, and birth defects. Babies can also suffer withdrawal symptoms after birth. Obesity increases the risk of diabetes, high blood pressure, and complications during delivery. It also raises the baby's chances of obesity and health issues later in their life. Make healthy choices, eat well, stay active, and avoid harmful substances. Thanks.